The G protein-linked receptor is a transmembrane protein that converts extracellular signals into cellular responses and spans the membrane 7 times with 7 alpha helixes. The receptor works with the G protein made up of an alpha, beta, and gamma subunit along with the GDP molecule attached to the alpha subunit. An extracellular signal binds with the receptor, which results in a conformational change of the receptor and an activation of the G protein. The GDP molecule that was attached to the alpha subunit is now replaced for a GTP molecule. GTP allows for the alpha subunit to move away from the beta-gamma dimer. The alpha subunit and the beta-gamma dimer can now target proteins to create a cellular response.
Up next are receptor tyrosine kinases. They are transmembrane cell surface proteins that act as signal transducers. They regulate essential cellular processes like proliferation, apoptosis, differentiation, and metabolism. First, signal molecules bind to the binding sites on the receptors. After this, the two receptor tyrosine kinases will move closer together to form an unphosphorylated dimer. After this, ATP will come in and phosphorylation will occur. This will leave one ADP and one phosphate group. This phosphate group will bind to the tyrosine group. This process is repeated 6 times, resulting in a phosphorylated dimer. The tyrosine regions become activated and act as kinase. Kinase is an enzyme that catalyzes the transfer of phosphate groups. After this, relay proteins will come in it and attach to the phosphate groups. The relay protein will change shape and will lead to its own signal transduction pathway. This will lead to different cellular responses based on the type of signal molecule that binds to the receptor.
The ligand-gated ion channel is a transmembrane protein that contains a pore which allows for the regulated flow of selected ions across the membrane. Extracellular ligands bind to the receptor, causing the protein to open up. Once the protein is open, specific ions are allowed to flow into the cell, which can produce a cellular response. As long as the protein remains open through the ligand, specific ions such as sodium, potassium, and calcium are allowed to flow in and out of the cell.